grip on it in order to move the field forward and to be able to do, which is a long dream of computational biology, to fold proteins. And one of the key problems we have with our present calculations is that we don't account for what is called atomic polarizability. You see here a bucket of water molecules moving around on the right side, the oxygen red, the hydrogen's white, and you see in addition a green charge, and that is a charge that describes how the different water molecules affect each other electrostatically and thereby polarize each other. And the degree of polarization, the direction is shown by this green charge. And this was considered a very difficult problem to compute, but you see on the bottom left that we actually achieved it. Again, we have a logarithmic scale, and you see that on parallel computers we can calculate polarizable and non-polarizable water molecules. We need about a factor of 2 to calculate the polarizable water. And that is, of course, good news. We can calculate it now, but it is a factor of 2 more. And if you calculate something for 2 months or for 4 months, it makes a big difference, and particularly if you have to do it many times. And so our idea is now to put that difference between red and blue onto the GPU, so that basically you do not have to pay the wall clock time price, but the GPU is doing it to you for free and rather cost effectively. Now, these were my main examples, but GPU computing is infectious itself. And my wife is also a professor at the University of Illinois, and she gave this morning in Venice, Italy, a lecture on using GPUs in a totally new field that didn't exist before because the computing was just too tremendous by any other means. And that is computing entire cells. Here you see how the response of E. coli cells when the following happens. Imagine that you have some E. coli cells, you cannot prevent it, on your kitchen table, and you have it very clean. Then the E. coli cells are sort of dormant. But now, as you see that on the left side, when you spill milk, then the milk molecules, lactose molecules, make it into the cell, tell the cell there's some delicious lactose around, and the cell now genetically begins to program itself to make proteins that import the lactose into the cell and catalytically degrade it into components that the cell is doing. Let me show it to you again. So here you see again how it works. And you see then each of the LAC-Y, they are called these green little spheres, proteins that are made by the cells. And you see it's rather stochastic, and the cells have actually personality. Some of them do it very quickly, some of them don't do it so quickly, and at the end you have actually 2 who are really outdoing all the other ones in responding and eating the milk most quickly. You see also in pink the messenger RNA that is the genetic message that is controlling this whole process. And then most important now for you as GPU programmers, you see on the left side the table with how effectively this calculation is done on the GPU, on an older model, on a newer model, and I'm supposed to tell you that this calculation was actually done on the cluster, on the Lincoln cluster at NCSA, where that was accelerated by the GPUs. Without the GPUs, this project wouldn't exist. With it, we have now a new field that permits us to simulate actually entire cells. So this leads me to the end of my lecture. And I just would like to add here that we are not only producing software, but we're also training in software. We have a long series of